on our way, and they'll find us there. I hope all of you are doing great. And I hope you enjoy this lesson. Okay, please, as this engagement takes place, make sure that you share your name and contact in the chat box. Okay, so type your name and your phone contact in the chat box. Okay, type your name there and your phone number in the chat box as we continue. So I hope you're doing well. Again, this lesson is brought to you by Yaaka Digital Network. It's one of our free packages right now that we are conducting that will be ending on 10th this month. And please, I encourage you to attend the online engagements that we have for you and your parents to learn more about digital learning. Someone left on the audio. Please turn off your audio. Okay, good job. I hope you encourage your parents also to attend those online engagements that we normally announce for them to attend, and you as well, to learn more about digital learning in this era, and to learn more about the opportunities that Yaaka has in store for you. So I encourage you to attend those engagements. They'll always be announced in your groups. Encourage your parents to attend too. Okay. Who can remind us on what we looked at the last time we met? Who remembers what we looked at the last time we met? Who remembers? What did we talk about the last time we met? Anyone wants to try? Right, Faith?
Practically, we talked about the cell theory and other parts of the animal cell, and then you say today we shall do into details.
Okay, good job. Yes, we looked at the cell theory, the origin of the cell theory. Okay, and then we saw some parts of animal cells, and then we say today we shall go into details. That is absolutely correct. And that's what we shall look at today, more details. Okay, and here they are. So this is the outer structure of cells. This is an animal cell, a eukaryotic cell in particular, and it is as viewed or observed under an electron microscope. Okay, an electron microscope. Once you view the animal cell, it will look like this. So now we shall look at some of the parts. So the parts of a typical animal cell, typically, this is what you'll find in any animal cell. The first glance, things that stand out are the cell membrane, the cytoplasm, cytosol, cell organelles, and cytoplasmic inclusions. Okay, so those are generally the parts of a typical animal cell. In case you observe a cell and it has probably one of these missing. For example, if it doesn't have the cell membrane or it has something else instead, that wouldn't be an animal cell. Okay, so these are the typical parts of an animal cell. And here is a picture of how that looks like. We have so many parts here we shall see as we move forward. The biggest picture, or I mean the most outstanding organelles here are the nucleus, of course, which also contains the nucleolus inside, and other parts like the nucleopore, which is an opening into the nucleus. We have heterochromatin, okay, or chromatin, and euchromatin, which all those make up the nucleus. All right. Here we have the cytoplasm, very, very outstanding. Okay. Have the cytoplasm over there. We have the centrioles. Okay, we shall see every part, and you guys are going to explain to me some of the functions. So be ready. See the Golgi complex or apparatus, vesicles, the ER or endoplasmic reticulum. This is the rough one, always surrounded with ribosomes. The smooth one is up here, smooth endoplasmic reticulum. And we have the mitochondria, okay, very many, and different other organelles, okay, there are a lot of them, and you're going to see some of them, and you guys will have to tell me their functions, okay. So here are some of them. Just like we've seen previously, we have the nucleus. Who can tell me the function of a nucleus? What is the function of a nucleus? Yes, Trevor?
Yeah, the nucleus controls all activities within a cell.
Okay, controls all the activities within the cell. That is correct. Any other? Yes, Elisha? Okay, Ian, you have something? Any other function of the nucleus, you know? Besides controlling everything else? Yes, Emma? Emma, unmute your audio and speak.
The nucleus contains cell organelles. It contains
Contains what? Okay, we can't hear Emma.
The nucleus contains nucleic acids, whereby we have the DNA and messenger RNA. So they are used mainly the DNA in protein synthesis, hence formation of other proteins.
Okay, very good.
I think it's a function.
Yes, it contains the genetic material, it stores the genetic material, which is good. All right, good job on the nucleus. Any other information you have on the nucleus, Faith?
Can I say it produces new chromosomes at cell division?
Okay, so the new chromosomes come from the genetic material. So when you say it contains the genetic material, that's exactly, that includes all that, okay? Even when the genetic material is divided, you form new cells that are individual. So that is included in just the nucleus having the genetic or storing the genetic material. Elisha, you have something?
Yeah, can I say that it contains the DNA which contains the may I say the codings for protein synthesis?
Yes, that is correct. It's the same thing as saying it contains the genetic material that is used in heredity, okay? Because the DNA that you see in the nucleus is the one that contains all the information that your cells inherit, okay? Every aspect of your body, that information comes from the DNA, which is stored by the nucleus. Okay, good job on the nucleus. Let's talk about the ribosomes. What is the use of ribosomes? Ribosomes, yes, Trevor?
Actually, the ribosomes, they help in decoding the message and the information of the peptide bonds.
Okay, very good. They facilitate, not even helping, because they don't help. They facilitate in decoding the information found in the DNA. So that is in protein synthesis. Therefore, we can just summarize that by saying that they facilitate protein synthesis, or they aid in protein synthesis, which is correct. Yes, Conrad, you're correct. In the chat box, you say they participate in protein synthesis. That is absolutely correct. What about the endoplasmic reticulum? Endoplasmic reticulum. Yes, Emma.
Well, the endoplasmic reticulum, can I speak about the smooth or the rough? Okay. Well,
Both,
okay,
if you don't mind.
In general, yes. Well, the rough endoplasmic reticulum is the one that contains okay, it is encrusted with ribosomes on the surface, on the outer surface of it, of the structure. And one function, we know that ribosomes are sites for protein synthesis. So, in other words, the rough endoplasmic reticulum is mainly for protein, okay, like for sites, it's a site for protein synthesis, was due to possession of the ribosomes encrusted on its surface, whereas the smooth endoplasmic reticulum, for it, does not contain any ribosomes on its surface. So it like not containing the ribosomes, it's mainly found in the liver, whereby it acts as a, like in the liver cells, where it acts as a detoxificant. Okay, like it's okay. I've messed up somewhere.
Okay, good job. No, good job. You didn't mess up. So that's correct. Also, especially on the rough, you nailed it, that it acts in protein synthesis because it is surrounded by ribosomes. Another one, let me see someone who has not spoken. Petra, tell us about the ER.
The ER can be used for lipid synthesis.
Mm-hmm.
Is it fine?
Yes, that is correct, Petra. The ER, especially the smooth one, acts in lipid and steroid synthesis. Oh, that's loud. Oh, okay, I've muted you. All right, someone was really having a bad echo. Yes, Petra, you're correct. Lipid synthesis and steroid synthesis is done in the ER. That is absolutely correct. I was saying something that is made up of steroids and lipids. Hormones. Hormones are made up, most hormones you find that they are made up of lipids and steroids. So ER aids in that. Good job. Any other thing about ER? Let me see someone whose hand is up. Faith.
I have formation of the Golgi body, especially by the rough ER.
Okay. Formation of the Golgi body, especially by the ER. Okay, maybe we'll elaborate on that. Jamie?
Excuse me, sir.
Yes.
I have a question about the previous point about synthesis of steroids. We talked about hormones. Does it mean hormones are an example of steroids?
Some hormones are made up of steroids, okay? Let's say an example would be the testosterone hormone. It's hard to pronounce sometimes. That's made up of fats or steroids, okay? Steroids are some type of fats. That's a very good example. So some hormones are made up of steroids, if that's what you're asking. Okay?
Yes, thank you.
You're welcome. Elisha, you have something? Yes, Elisha? Okay. Let us talk about the Golgi, not the Mitochondria. You went off. I picked you and you went off.
About the rough endoplasmic reticulum, I think it also facilitates the transportation of proteins that have been synthesized by the ribosomes.
Okay, that's correct. It also facilitates in their transportation. Good job. Okay, the mitochondria. Let's talk about the mitochondria. What is the function of the mitochondria? Who knows? The mitochondria. Yes, Emma.
Well, the mitochondria, we know that mitochondria
Yes,
itself. Well, the mitochondria, we know that it's a cell.
Someone has their audio on. I've muted them, I guess. Please, you turn off your audio if you're not speaking. Okay.
Well, well, the mitochondrium, we know that it's a self-autonomous organelle that is able to make more copies by itself. Like it can replace how the worn-out mitochondrial cells. So it being self-autonomous, it contains the DNA. Hence, because it's a site of production of ATP, energy release, whereby this energy is required by the body cells to function properly. So that's the main function, because if it loses function and like it has lost out shape, there will be less production of energy required for life processes. So
okay, good
that's
job.
the main function.
Yeah. So the function is exactly what you said, is energy production in form of ATP. And you have mentioned the key characteristic that we even looked at last time, that this organelle particularly contains its own DNA, its own circular DNA, which aids in its division, okay? Which aids in its division. So it's important to remember that. But the main function is energy production. Let's look at the Golgi apparatus or the Golgi complex. What is the function of the Golgi complex? James, Justice. James, Justice, or Justice James. Function of the Golgi apparatus. James, Justice, can you hear us? Okay, if James cannot hear us, Arthur, you can go.
Golgi complex receive the new synthesized proteins by ER via transfer vesicles.
And what does it do with them? Yes, Arthur. After receiving the proteins, okay, you're correct. It receives them, and then what it does is to continue processing those proteins. It can add carbohydrates to form glycoproteins or fold them into different structures. Okay, so that's its main function. Someone also mentioned in the chat box that the Golgi apparatus makes lysosomes, which is also correct. Lysosomes, and we shall see the function of lysosomes. Who else would like to talk more about the Golgi apparatus? Jamie, do you have any other thing?
Yeah, I would like to say that it replenishes the cell membrane by producing vesicles which become part of it after discharging the contents. Then,
yes,
it is concerned with the storage of and modification of lipids, plus it facilitates the secretion of glycoproteins and their processing, for example, the mucus, and then it accumulates and secretes. It accumulates and excretes some substances out of the cell, but I don't have the example of those substances.
Okay, good job. Yeah, that is correct. And the way it accumulates and expels those substances through forming of those lysosomes or vesicles. Those substances can range from toxins that enter the cell or bacteria or anything like that. So that is very, very good. Those are great functions of the Golgi apparatus. Good job. What is the function of lysosomes? Function of lysosomes. I need someone who has not spoken. Teddy. Yes, Teddy.
One function of a rhizome I can give is that they carry out digestion of materials taken in by endocytosis.
Okay, digestion of materials taken in by endocytosis, that is correct. Another one, who is here? Let me see, Arthur. Yes, Arthur, another function of lysosomes. Elisha, you want to try?
Yeah, sir. I think it also aids in the destruction of old cells to give space to rise of new cells.
Exactly, okay. It also facilitates in destruction of the old cells in order to give rise to the new cells. Very good. Those are good functions also. What about the microfilaments? What is the function of microfilaments? Microfilaments, yes, Irene. Irene. You can unmute your audio and then speak. Abenacho Irene. Swipe left, unmute. then talk.
Yeah.
Okay, I guess she hasn't figured that out yet. Let's have Petra.
The microfilaments contain catalase that break down hydrogen peroxide, which are associated with oxidation reactions.
Okay, good job. We see microfilaments doing that. And what is their main function besides that? Who would like to try? Let's have Faith.
The microfilaments take part in cell division.
Okay, they aid in cell division. Yes, that is one of their functions. We see them doing that also. What else is the main one that we see? Yes, Elisha.
I think they also provide a cytoskeleton to the cell.
So that's the main function of these microfilaments. They act as the skeleton for the cell. So they are the cytoskeleton you hear about, okay? Strengthening the cell. Yeah, strengthening the cell not to break apart. Every time you hear cytoskeleton, we're actually talking about the microfilaments, and sometimes you also have microtubules in there, okay? They are for cell strength. That's their main function. Okay, good job. I think Petra is looking for something. Let me mute you. Okay. Cleavage of animal cells. All right. Centrioles. Where do we see centrioles exerting their purpose in the cell? Yes, Emma? Emma? Function of the centrioles? Okay, Emma cannot unmute his audio to speak. Let's have someone else. Let us have Diana. Yes, Diana.
They support the cell membrane.
Supporting the cell membrane.
They support the cell membrane.
Supporting the cell membrane, okay. What else? What is their main function? Trevor.
The main function is that they help in cell division in the animal cells.
Yes, the main function is formation of the spindle fibers that aid in cell division. So Jamie nailed it very well. They aid in formation of the spindle fibers during cell division. Okay, good job. Microvilli, the function of microvilli. Microvilli knows the function. Yes, Dan. Okay, Precious.
Microvilli.
Precious.
They provide a large surface area for absorption and digestion.
Okay, very good. The microvilli will always provide the largest surface area for absorption. Okay, absorption. So instead of saying digestion or anything, just say absorption because they're found just outside the cell in this case. All right, cilia and flagella will be our last one. What is the function of cilia and flagella? Okay, if a cell contains cilia or flagella, what does it use it for? Yes, Richard? Richard?
They are used for movement.
Okay, movement of the cell. That is correct. All right. So those are the functions of the parts of the cell organelles. They're very, very important for us to know. The microtubules, we looked at them together with the microfilaments. That's why I've skipped them. So that's good. We also see cytoplasmic inclusions like the pinocytotic vessels or vesicles and the glycogen granules. The pinocytotic vesicles, they act in pinocytosis, which is the other name for pinocytosis is what? Who knows the other name for pinocytosis? What's the other name for pinocytosis? Yes, Elisha?
Cell drinking.
Yes, cell drinking, okay? That's pinocytosis. So those vesicles help in cell drinking. Glycogen granules, storage of glycogen, which is the storage form for glucose, and then fat droplets for fat storage, which is great. So those are the functions of the different parts of the cell. Any questions so far? Any questions here? Sophie, do you have a question?
Yes, master. Like I was kind of asking to like help me out with like a difference between pinocytosis and phagocytosis.
Okay, who can differentiate for us pinocytosis from phagocytosis? Who would love to explain that? Anyone? Richard, go ahead.
Pinocytosis is whereby the cell takes in fluids like water, cell drinking, while phagocytosis is whereby the cell takes in large substances that are in solid state.
Okay, very good. So pinocytosis is cell drinking, phagocytosis is cell eating. In pinocytosis, substances that are taken in are liquid in nature. In phagocytosis, substances that are taken in are solid in nature. That's the difference. Okay, so very good, Richard. Good question also. All right.
No.
Any other questions? Any other questions? Richard, you still have your hand up. Is that a question? Okay, your hand is down. Good. So those are the parts and their functions in an animal cell. So we have this kid here saying that, so the vacuole basically stores its own diseases. Wonderful. So you see these vacuoles will store anything. All right. Let's look at the animal, the plant cell. Okay, the plant cell. Here are the parts of the plant cell. We're going to look for the parts that we do not talk about in the animal cell, and we shall explain those. The first part that I see here is the cell wall. Okay, so what is the function of the cell wall? Function of the cell wall? Yes, Jamie?
It provides mechanical support and skeletal support for the cells as a whole. Then the cell wall is fairly rigid and resistant to expansion, and so by that it allows development of turgidity when water enters them osmotically. Okay. The cell wall also it's modified as food reserves, for example, as in storage of hemicellulose in some seeds of some plants. Then it transfers, the cell wall, it develops a coating of waxy cutin, which is called the cuticle, on the exposed epidermal surfaces, reducing water loss and the risk of infection in the plant.
Okay, very
That's
good.
what
Those are good answers on the function of the cell wall. Well done. So we see mainly that it provides it maintains the structure of the cell, so the cell does not collapse because of the cell wall. Also protecting it, right, from various entries. And we also see the secretion of the cuticle in the epidermis cells and also other functions. So good job. Autophagy and autolysis as functions of lysosomes. Okay, those ones you mentioned them. So please, if anyone finds time, read the comment by, I think it's Tiaba. All right. So those are good functions of the cell wall. Plasmodesmata. What is the function of plasmodesmata? Function of the plasmodesmata. Who would love to try? Besides Jamie, who would love to try? Richard.
Plasmodesmata help in the transporting of substances from one cell to the other.
Okay, allows in substances from one cell to another. Wesley, do you have something? Wesley? Okay. All right. Yeah, transportation or movement of, they allow movement of substances from one cell to another. That's good for plasmodesmata. Which, someone has a hand up. Melissa, you have a question?
What I think plasmodesmata. No, I'm adding to the plasmodesmata function, and I think it's a site for active transport.
Very good. Also aids in active transport. So that's still transportation of materials from one cell to another. Good job. The other part we can look at is the cytosol. What is the function of the cytosol? Someone is asking, how does it do it? The plasmodesmata, you realize it's an opening. It's right here, okay? So if it is an opening, meaning it's gonna allow things to pass through it during active transport, okay? It's right there, I've circled it. It's an opening into the cell wall, through the cell wall into the cell. Cytosol. Functions of cytosol. Okay, Jamie.
all I know is that the cytosol is important for the biochemical processes. Okay, like it's involved in the movement of materials by streaming and at times locomotion of unicellular organisms.
Okay, good job. Good job. Nice. As mainly part of the cytoplasm that allows those chemical processes, and also in other organisms, it will aid in various functions like movement, especially in single cellular eukaryotes like Paramecium. Okay, good job. Let's look at another organelle, which is the chloroplast. I think it's the only one that is here that we have not looked at. Chloroplast, what is the function of the chloroplast? Function of the chloroplast, quickly, Emma. Or Sharon, you can go, because Emma, every time I pick him, he doesn't talk. He has failed to unmute his volume. Oh, this time he's successful.
The chloroplast contains
a pigment called chlorophyll that is used to trap sunlight, that is used during photosynthesis.
Okay, it's a site of photosynthesis. Very correct. Good job. Site of photosynthesis. So now we know all the parts of the cell and their functions. All right, good job. Any questions? Okay, if there's no any questions, maybe Faith has a question.
I wanted to add on the function of the chloroplast.
Okay, please go ahead.
Can I also say that it makes ATP during the light stage of photosynthesis, the process known as photophosphorylation.
Okay, that is correct. During the light process, it makes ATP, but that ATP is later used in the is utilized in the dark reactions, right? Later.
Yes.
Hmm? Is it not so?
Yes.
Okay. Good job. James Justice, you have a question?
Yes, sir.
Hmm.
I'm asking the difference.
We can't hear you. Type the question in the chat box. Your network. We can't hear you well. Okay, so someone is asking here, is a cell wall a prison? Is it a cell wall or just a prison? Because we know that the cell wall won't allow things to pass through easily. So most things pass through by active transport. It is sort of a prison for things in the plant cell. All right, let's look at the comparison between the two cells, plant cell and animal cells. And this will be quick. Just you look at the feature and if it is present or absent. Let's start with the cell wall. We see that the cell wall is absent in animal cells and is present in plant cells and is made up of cellulose. Okay, plastids. Plastids are absent in the animal cell and present in the plant cell. An example of a plastid is chloroplast. Okay. We have the plasmodesmata, which we have explained. It is absent in animal cells since we lack a cell wall, but present in the plant cells. Okay. Remember, you guys, to share your contacts in the chat box. Only a few shared and the rest have not shared. Write your name in the chat box and your phone number as we continue. We have cilia. Cilia is present in some animal cells, but most plant cells will lack cilia. Okay. We have centrioles that we have looked at. They are present in the cytoplasm of animal cells, but absent in plant cells. Okay. All right. Cholesterol in the cell membrane, we have it present and absent in plant cells. And then I repeated centrioles. Okay, we finished that. Vesicles, they are present in animal cells, absent in plant cells. The shape, irregular, and then a fixed shape in plant cells due to the cell wall. The vacuole, very small if present, and then the vacuole in the plant cells is huge, takes up 90% of the cell volume. Then the food stored in animal cells is glycogen, and in plant cells it is starch. So that's very, very important. Those are the differences. Okay, good job in those sharing their contacts. So those are the differences. We can look at the similarities. I have a picture of a cell here. Just give me 3 similarities only. 3 similarities. You can raise up your hand and give us any 3 similarities. Yes, Sharon?
Like in both plant cells and animal cells, they all have the mitochondria.
They both have the mitochondria, correct? Elizabeth?
Both have the endoplasmic reticulum.
Okay, they both have ER. Teddy?
They both have the cell membrane.
Cell membrane, correct. So here are other similarities. Good job. And the rest of the similarities are right here. So we have the cytoplasm, the ER, ribosomes, mitochondria, Golgi apparatus, microtubules and microfilaments, nucleus, lipid droplets, and many others. So those are the similarities. Good job. Okay. Well done. Good participation. All right. This is a short story of these 2 guys. This guy told the other one, wow, you look stacked. And then this one was like, thanks, I've been working on my thylakoids. See, they look like abs. Eh? They look like they have a 6-pack. This is like a 20-pack. So this guy has been working hard to impress other thylakoids. Let's look at the cell membrane in a minute, and then we'll continue from there next time. The structure of the cell membrane. It is described as fluid. These are the parts, okay? We have the glycoproteins, carbohydrates, glycolipids. We have the integral proteins, peripheral proteins, those don't cut through. Then channel proteins that act in active transport. Then the alpha helix of protein. That's another type of protein that is found in the cell membrane. We also have the globular proteins over there. So first of all, it is described as fluid. Why is it fluid? Who knows? Why is it described as fluid?
Because
see, who knows? Who knows? Yes, Sharon. Contains what?
Because it contains the glycolipids.
Contains glycolipids. Okay, good job. Another one, Teddy. Yes, Teddy. Okay, Melissa. Yes, Melissa.
It's fluid because it contains it's fluid because it's made of fatty acid tails, I mean phospholipids that have unsaturated tails that are not closely packed together that makes it flexible.
Okay, good job. That's good. All right. So we see that these guys, let me first meet this guy. Where is this guy? Okay, done muting him. So according to C.J. Singer and G. Nicholson, 1972, the structure of the cell membrane is a fluid mosaic model. And the reason why it is fluid is because the individual phospholipid and protein molecules can move laterally, giving the membrane flexible structure that constantly changes shape. Okay, this is exactly what you've said. And why is it mosaic? It's because the proteins that are embedded in the phospholipid layer or bilayer vary in size, shape, pattern, and arrangement. That's why it is mosaic. Okay, that difference there makes it mosaic, and that's why we call it a fluid mosaic model. Okay, the main components of the cell membrane are phospholipids, proteins, carbohydrates, and cholesterol. Okay, and the description of the fluid mosaic model is here. It is 2 layers of phospholipids whose lipid tails face inwards of the membrane, while the heads face outside. The phosphate heads are polar, which means they are hydrophilic. They love water and form hydrogen bonds with water. Lipid tails are non-polar, hydrophobic, and are attracted to each other, which is why they form the van der Waals forces. So here is the structure. The heads are these ones. Okay, they face outside and they are hydrophilic, but the tails are hydrophobic and they face each other. All right, they face each other. These are the tails. So that makes it. It's also one of the reasons that we have said it makes it fluid. Okay, the layers are able to move a little bit around each other. Okay, so that is the description. And to add more the description, we see that the intrinsic proteins are found at the inner and outer surface. So these, and then we have the ones that are in the outer surface and also the ones that are in the inner surface. Some intrinsic proteins are partly embedded in any one of the phospholipid layers. So some are partly embedded, some go all the way through. Some transmembrane proteins are porous, like these ones, which we call the transport proteins. This one right here. And then some phospholipids conjugate with short branched carbohydrates to form glycolipids. I've seen glycolipids, which are here. I mean glycoproteins, not glycolipids, glycoproteins. The glycoproteins are, we have a carbohydrate attached to a protein, right? That makes a glycoprotein. In animal cells, cholesterol molecules squeeze between the phospholipids. And you can see some cholesterol here, these yellow ones. They squeeze in there, right? Remember, the cell membrane is supported by the microfilaments that we talked about. Here are the functions. You go through these functions of the cell membrane. We have talked about many of them when we were talking about the cell. So you can always go through them. You can see the general one is protective barrier between inside and outside the cell and determines the cell shape and size. Okay. On the proteins part, we see so many functions of proteins, especially in active transport. On the lipid side, we see cell-to-cell recognition. Sometimes when this is affected, you find that people develop autoimmune diseases. Some cells cannot recognize other cells in your body. They end up eating each other seriously. But the glycolipids help in cell-to-cell recognition. Cholesterol stabilizes the membrane structure for preventing phospholipids from closely packing together. And then the bilayer, which is semi-permeable, controls the movement of substances in and out of the cell. So yeah, that's the description and the function of the cell membrane. I'm going to send out these notes for you to enjoy going through them. Here is a structure showing the different types of proteins. These two structures show those that are halfly embedded, those that allow transportation, those that are in the lower side or the inner side. And that was the function of that picture. Show you the different types. Lastly, this is how every other organelle looks at the mitochondria, since it is the powerhouse of the cell. Every organelle admires it, seriously. That's the picture that shows how important the mitochondria is. Well done. Let's come back for chemistry, and I'll explain this in the chemistry class. So well done. See you in chemistry. just take a break of 5 minutes, come back for chemistry. And if you have questions, remember to type them in the WhatsApp group, and we can look at them